about designing controlled experiments to investigate cognitive and behavioral processes using animal model. So the first slide basically tells the various factors that can affect the results of an animal experimental studies. So the factors includes the state and health of experimental animals, the test conditions, the level of measurement, data acquisition, level of automation, technical state and calibration of test equipment. Then there are different other factors, trait factors, which involves the strain, subline, genetic background, gender and age of the model organisms. The time of testing, that is day, night or whatever time of the day that you are testing the doing a particular test. Then the experience of the person that is performing the experiments, and the place where the animals have been kept, and the development conditions like housing conditions, social hierarchy. These all are various factors that affect the results of an animal experimental study. So what's the difference between cognitive versus behavioral processes? So when we do behavioral analysis, we can test two different type of parameters, and in those two parameters as well we have various other various tests that we can perform for cognitive evaluations as well as for general behavior evaluations. In cognitive evaluations basically involve the mental activities of the brain, such as learning, memory, attention, perception, language and decision making. They help an organism understand and respond to its environment. So cognitive evaluations basically tell us about the ability of an animal to process in a particular environment, how the brain of an animal thinks. The behavioral processes are the observable actions or responses that result from these mental activities, like exploring, eating, avoiding danger or social interaction. So the behavioral processes tells us about how the body of the animal acts based on the thinking. So these are the various model organisms that you have already been sensitized about in previous lectures by other dignitaries. So there are different model organisms like C. elegans, Drosophila melanogaster, mouse, rat and monkeys. And the other image on the side that basically shows that how amyloid beta accumulation and plaque formation leads to cognitive and motor alterations, and how in the preceding slides we will see that how these motor and cognitive alterations can be tested in various neurodegenerative disease animal models. So rats and mice are among the most commonly used animal models in behavioral neuroscience research. The basic reason is that it is easier to evaluate the behavioral changes in rodents rather than other model organisms like C. elegans or Drosophila melanogaster, because you cannot detect various different changes in their general behavior or cognitive evaluations in other model organisms, but it is rather easier in rodents such as rats and mice. From tests of basic locomotor and sensory functions to analysis of more complex behavior related to cognition and emotionality, various types of behavioral tests are being conducted on rodents. These behavioral tests exist for various neuropsychiatric disorders like anxiety, depression, schizophrenia, autism, addiction, ADHD, post-traumatic stress disorder, and neurodegenerative diseases like PD, AD, Huntington, stroke, etc. Okay, so this slide tells us about the behavioral paradigm. Structured experimental setup. So behavioral paradigm is basically the setup, experimental setup that is used to study specific behaviors under controlled conditions, which helps link brain function and observable actions. Its purpose is to model human psychological or neurological disorders and assess learning, memory, anxiety, pain, motivation, and decision making. So these tests help in drug testing, evaluating the effects of a particular drug, how they affect the brain of the animals, and studying disease mechanisms. Like if a drug affects the cognitive ability of an animal, you can ascertain that based on which type of cognitive ability is being compromised, like it is memory, in memory it is working memory or recognition memory, what kind of memory is being affected in that particular disorder. You can ascertain the underlying molecular mechanisms, and the validation of that will be done through the molecular assays that you will be doing on in the ex vivo part after isolating the brain. So okay, so it also, these tests are also helpful in telling that how genetic or environmental factors affect behavior, and the common examples of these behavioral tests that are being widely used in animal experimental studies are open field tests, which are used to evaluate the locomotion and anxiety levels. Then there is elevated plus maze for anxiety behavior, Morris water maze for learning and memory, tail flick and hot plate test for pain response. So we'll start with the locomotor tests. So locomotor tests basically assess the movement, activity levels, and motor coordination in animals. These are often used to evaluate the effect of drugs, neurological disorders, or genetic modifications, and can detect hyperactivity, hypoactivity, or motor deficits. Common examples are open field tests. So in open field tests, there's a large open square arena where you leave the animal to explore the arena, and you check the basic behavior of an animal. Since you know that rodents are, their natural behavior is to explore a particular area when they are left in a new environment. So if there are any alterations from that behavior, they are not exploring. There are several different aspects or parameters that we study in exploring. One of them is rearing. They stand on two paws, and their upper two paws are in the air, and they are exploring all the walls, and they are checking all the things that are in that square arena. They sniff the odor, a particular odor. They explore the arena. So if there is an alteration in that particular behavior of an animal, the animal is not exploring, is showing anxiety like behavior. How will an animal show an anxiety like behavior? If the animal is not exploring, we have left the animal in the arena, but it has gone from its origin point where we have dropped it to some corner, and it sat there for the whole entire testing period, and it is not exploring the arena. So we know that there is some kind of anxiety, or if there is hyperactivity, the animal is exploring more than expected. So how you will compare that? You compare that from the control rats. You, every time you do certain tests, you use control rats as well as diseased rats. Then you compare the results of the diseased rats in comparison to the control rats. Your standard baseline will be the behavior of control rats. So the other test, there's rotarod test, which is used for assessing the motor coordination and balance by timing how long an animal stays on a rotating rod. So by time the speed of the rod, we keep on increasing the speed of the rod. We first sensitize the animal of how to walk on the rotating rod by making it walk on a slower speed, and then we gradually keep on increasing the speed to make the animal learn the process of balancing on the rotating rod. So if the animal learns well to maintain the balance in the repeated experiments where we keep on increasing the speeds, so that means that the animal's motor coordination and balance is Okay, but in other case, when there are they are compromised in such in movement disorders such as Parkinson's, these motor coordination and balance parameters are compromised. So in that case, we will see that how much we try that the animal stays on the rod, but it will keep falling down. You will try again and again, but the animal will keep falling down, and this tells us that the motor coordination is impaired in diseased rats. Okay, then third is wheel running. So these are just the examples, and there are various other tests that are behavioral tests that we'll be discussing in next slides. Wheel running basically tells voluntary activity measured over time to assess motivation and endurance. So the animal is left in a rotating wheel, inside a rotating wheel, and we check that how long the animal is moving and how fast the animal is moving. So the speed, balance and motor coordination are again the things that we test in wheel running experiments. Okay, so this slide shows the setup of an open field test. This is the most basic and common test that is being done for any kind of animal experiments that people do concerning brain, obviously. So this test basically tells us about the normal behavior of an animal, the exploratory behavior. The purpose of the test is to assess spontaneous locomotion and anxiety levels. The procedure is that the rodent is placed in a large enclosed arena and is left to explore the arena to test the exploratory activities of the open field test. And the applications of this test are to check the drug effects, genetic model assessments. So these are the various behavioral parameters that we can study using the open field test. So there's locomotor activity, track plot pattern, center zone entries, time spent in center. So as I already told that more the animal stays in the center of the arena, so it tells us about that the animal, the anxiety is less in that animal. If the animal is exploring, it's staying in the arena, it's going in and out. So that tells us about the absence of anxiety in those animals. So if you see the other columns, you can see that there is a comparison between control rat and PD rat, that how these parameters will be differing in different model rats, disease model rats and control rats, and what is the interpretation for that, what those changes tell us about. So if you see the locomotor activity is high. There is extensive movement across the arena because the animal is exploring the arena properly in control rat, but in comparison you can see that in PD rat there is low restricted movement which is often confined to the corners. So this indicates bradykinesia and reduced exploratory drive. Bradykinesia is basically the slowness of the movement of a rodent. Then we can see the track plot pattern. The track plot pattern is dense, and there is uniform coverage of center and periphery in the control rats, but it is opposite in rotanon or PD rat model. So all these observations are basically based on the test that we have ourselves carried out in our laboratory, and I'll be sharing the videos of the same with you towards the end of the presentation. So I'll be showing you the videos of the control as well as the PD model rats and how the behavior changes happen between them, the differences between their behavior parameters. Moving on, this is the overview of rotarod test. So it is used to evaluate rodents' motor skill, learning abilities, and the procedure is that the rodent is placed on a rod that accelerates, and the rotating rod elevated above padded base. So this is just the setup and applications of rotarod test. Applications are relevant for Parkinson's and ataxia research. Then next test is beam walking test. Its purpose is to evaluate rodents' fine motor coordination and balance. In this, the rodent traverses a narrow elevated beam, and there's a narrow beam which is suspended between the platforms, which basically is used to test the motor coordination and balance. So the applications involves the cerebral function studies and neurodegenerative research like Parkinson's disease. Okay, so these are the different behavioral tests for sensory perception. These involve hot plate, one fray, and tail flick. So why sensory perception is important to study? So it tells us about the animal's complete ability to detect external stimuli, process the information through neural pathways, and generate appropriate behavior responses. In pain research, we focus specifically on nociception, which is the neural process of processing noxious and detecting pain stimuli. And why behavioral testing matters? Because behavioral test serves as an essential tool in controlled laboratory environment, and they provide quantifiable reproducible metrics that bridge molecular mechanism with whole organism responses, enabling researchers to advance fundamental understanding of pain pathways and modulation, screen novel analgesic compounds with clinical potential, and validate therapeutic targets in disease relevant models. So the first test is hot plate test, thermal nociception. This test basically evaluates an animal's integrated response to sustained thermal stimulation. A rodent is placed on a precisely controlled metal surface maintained at 50 to 55 degrees Celsius, and the researchers measures the latency period until specific pain-related behavior emerge. And what are those behaviors that are observed are paw licking or shaking, jumping or escape attempts, vocalization. So vocalization is basically species dependent, and a mandatory cutoff time of typically 30 to 60 seconds prevents tissue injury while maintaining ethical standards. So this is what is kept in mind while doing these tests. So this test primarily engages central pain processing pathways requiring integration across multiple levels. The second test is one-fray test. This employs calibrated nylon monofilaments of increasing stiffness. So pain in the form of, you can say, a needle is pricked to the paw of an animal. And the stiffness of that particular needle keeps on increasing with the passing time. We start with a small pressure, but then we keep on increasing in the following tests. So this is used to quantify the mechanical nociceptive thresholds. Each filament delivers a known force measured in grams. So when applied perpendicular to the plantar paw surface until buckling occurs. So the 50% withdrawal threshold is calculated using the up-down method and providing a statistically robust measure of mechanical sensitivity. The clinical relevance of this particular test is in neuropathic pain conditions. Example, diabetic neuropathy, nerve injury, mechanical allodynia, which is a type of nerve pain, and the inflammatory hyperalgesia following tissue injury. The test's quantitative precision makes it ideal for dose response curves and longitudinal studies tracking sensory changes over time. The third test is tail flick test. So in this, a focused beam of radiant heat is applied to the tail of the animal, and the latency to reflexive tail withdrawal is precisely timed. So this is also a pain evaluation test. So the applications of this involves rapid assessment. Advantages and applications involved. First is rapid assessment. Tests can be completed in seconds with minimal handling stress. Second is reflex specificity. Primarily test spinal circuits useful for distinguishing central versus peripheral analgesic effects. The third is reproducibility. There's less variability than tests requiring complex behavioral integration. And the safety measures. There's an automatic cutoff, typically 10 to 15 seconds, which prevents tail tissue damage. This test is particularly useful when studying opioid analgesics as it can differentiate spinal from supraspinal mechanisms of action. Okay, so how to select a particular test for our research that we will be doing. So there are several parameters to consider. So selecting the appropriate assay, it depends on the research question, what you want to study, the neural pathways of interest and the pain modality being studied. Different tests have different applications and different uses. Like hot plate test is particularly useful if you want to check the do the opioid screening or central pain processing. One fray test for neuropathic pain and allodynia studies. The tail flick test is important if you want to screen analgesics and spinal mechanisms. Okay, this is just a further extension of the applications and impact. The first one involves the fundamental pain neuroscience. Elucidating the molecular and cellular mechanisms underlying acute and chronic pain states. So these assays enable the researchers to map nociceptive pathways from peripheral transduction through spinal integration to cortical perception. The second is analgesic drug discovery, so screening novel therapeutics and their effect in reducing the pain, or maybe if they're increasing the pain, to induce the pain, what kind of effect the drugs, novel drugs are showing. The third is disease modeling studies, characterizing sensory abnormalities in models of neuropathic, inflammatory, cancer related and post surgical pain. Longitudinal testing tracks disease progression and therapeutic responses over time. The last is neural circuit analysis, dissecting the specific contributions of peripheral nerves, dorsal root ganglia, spinal cord circuits, and supraspinal centers. Combined with optogenetics and chemogenetics, these tests reveal causal circuit behavior relationships. Then first we saw about the motor coordination test, normal anxiety exploratory test. Then we saw about the pain related tests. Now these are the learning and memory function tests. The first one we'll be discussing is Morris-Water maze. So in this, a rat or a rodent is basically left in an open water tank to explore the arena. And there's a hidden platform in the open water tank which we want the rodent to identify. And the first time we place the rat in that arena, the rat takes longer to find the hidden platform, because why it finds the hidden platform is because it has to, otherwise it has to keep swimming for that duration. Therefore it finds the platform to go into the resting phase. So the first time when you place the rat in this water tank, the rat takes longer to find the place where the hidden platform is. But in the preceding experiments, when you again put the same rat in the same conditions, you will see that the rat has developed a memory that where that platform is, and it will straight away swim to that particular place to reach the hidden platform. So this tells us about the that the memory impairment in this particular rat is not there, and this is a control rat. So if you want to study the learning and memory, this test plays an important function, and this can be useful in the study of neurological disorders like with cognitive components such as schizophrenia and Alzheimer's disease. So I've already told you the procedure for this. So this image basically shows us the phases that I have told. In the first phase, you can see that the rat takes, we have dropped the rat here. This is the origin point. The rat takes longer to find out the hidden platform, which might be here or here, right? In the second test, we have dropped the rat here, and it straight away goes to that particular place where the hidden platform is. The third one shows impaired memory function in a rat. So the rat is exploring the arena, but it is not able to find the hidden platform. Okay. So the next test for testing the spatial learning and working memory is radial arm maze. So in this, there's a central platform and there are 8 arms around it, which is used to assess the working memory. As in Morris Water maze, cues are placed around the room. There are different cues placed in different arms of this maze to distinguish for the animal to distinguish between different locations. That the animal will know that there's a particular reward placed in one of the arms when we placed for the first time. The animal will explore completely and it will know that in these particular arms there is reward placed for it, and the next time you keep the animal in that, it will develop a memory and it will straight away go to that point where the reward has been placed for the animal. So okay, the third one is Y maze test. Like a radial arm maze, there were 8 different arms. In this one there are three arms. Okay, so in this test is basically used to evaluate the spontaneous alteration behavior in rats. And in this, the rat, for the first time the rat is placed in an origin arm, as you can see here, and the animal is allowed to explore the arena. All 3 arms are open for exploration. In the second phase, when you will keep the same rat here, you will block one arm and you will allow the animal to explore the 2 open arms. So since the normal behavior of animals is exploratory behavior, they explore the novel areas where they are placed. So when you in the third phase, you will again open this arm and you will see the frequency with which the rat is visiting this novel arm, because for the rat, this arm is the area that the rat has not explored. Right, so the frequency of entering the novel arm will be more in comparison to the other arms if the memory is not impaired. So this is Y maze test. Next is novel object recognition. This is again a memory evaluation test. So in this, what we do is, in the first phase, we place the rat in an open arena. The arena is similar to that of open field test. There's a square open arena where we place the rat, and there are 2 similar objects placed in the arena. The animal will explore both the objects in this phase, and in the next phase, what we do is we change one of the objects with a novel object. So we see the ability of the rat, or the time the rat will spend with the novel object, because of its normal exploratory behavior. The rodents possess. So if the rat's memory is not impaired, it will spend more time with the novel object rather than the similar object. If it has gained a memory that I have already explored this one, it will not spend more time with that, but with the other one. So this is another memory test. These are the various parameters that will be evaluated using the novel object recognition test. The first one is the exploration duration, which is high in control rats because it actively explores both the objects, and in PD animals it will be low, which tells us that there's reduced motivation and cognitive impairment in those rats. The object preference in control rats will be that the rat will be spending more time with novel object rather than the similar or the familiar object. But in PD rats, there will be brief approach to familiar object only. These results are again based on the results that the experiments that we have done in our lab, and I'll show you the videos for it. So these are all the observations for control rats and PD model rats. So there was a brief approach to the familiar object in case of PD rats, but however in the control rats, they explored both the objects but spent more time with the novel object. So there are various interpretations based on it, which tells us that In PD rats, we saw impaired recognition memory, bradykinesia, reduced exploratory drive, and cognitive decline, motor impairment, etc. Then there are tests for anxiety and depression-like behaviors. The first one is forced swim test. So in this, this test basically is helpful in identifying new classes of antidepressants and anxiolytic drugs, as well as discovering genetic factors linked to mood disorders. The test uses immobility as a measure of behavioral despair. So the procedure involves the animal is placed in a cylinder filled with lukewarm water. A normal animal will initially display swimming and escape behavior, but will gradually spend increased time immobile, that is floating. However, a diseased rat will show more anxiety-like behavior as it will keep trying to escape the cylinder by showing climbing like behavior. It will keep climbing and try to escape, and there will be more anxiety in the diseased rats as compared to the control rats. So okay, so that was all about the different kind of behavior test, and now we'll be talking about a software. This is Anime's behavioral tracking software, which is basically used to test the behavior of animals. This okay, so most of the people do the behavior test manually. So there are various variety of alterations that a manual test can give. It could be based on the person that is performing the test. Okay, so there could be various different changes than the normal behavior of the animal that you can see in a manual test. However, the Animaze is an automated software that can give us the actual behavior of the animals that they are displaying, and we can test at a particular time. If we are performing one test, we can get a number of different parameters. For example, if you are doing open field test, you place the rat in the open arena for around 3 minutes or 5 minutes. In the normal manual test, you will only see if how many times the animal is rearing, how much time it is immobile, that is it has not moved, how much time it is taking to explore. Okay, so but in when you use these behavior tracking softwares, you can get the precise number in seconds, time in seconds, that how much time the animal took to explore the arena, the number of in and outs, like it went to the center and then came out, how much time it spent in the corners of the arena. You can basically make different grids, specify different zones to test that in one particular zone how much time the animal spent. In different zones, you can compare the experimental parameters in between different zones in one single test only. So its purpose is to automatically record and analyze animal movement, interactions, and behavioral metrics. It is widely used in rodent studies to test different behavioral assays like open field, Y-maze, and novel object, etc. The core features of Animaze involves flexible video tracking, works with the live camera feed or pre-recorded videos. So you can use both. You can either record the videos live, or you can feed pre-recorded videos as well. Supports many camera types. There are simultaneous multi-camera option as well available as well. You can track the whole body or head or tail alone, or you can do the partial tracking as well as whole body tracking. So as I already told, you can define different zones in the apparatus, and you can test speed, immobility periods, path efficiency, head orientation, rotations, etc. You can do manual scoring or take the notes from the automated scoring that you will the data you will receive after the test that you are doing. These are the various applications. You can use anime software for all different kinds of behavior tests like open field tests, Y-maze, NOR, anxiety, lighten, dark box, elevated plus maze, water maze, specialized assays, etc. Okay, so this is a little video of anime software, how to use this software. I'm sorry. So this part is basically defining the walls of your apparatus. You are defining the walls in between where you want to do the analysis. Okay, so using anime software is also beneficial for publication purpose, because when you do manual behavior testing, you have recorded the data yourself. You don't have any proof that you did this particular test or how the results were. Have you manipulated the data or not? But in this case, you have recorded validation for your results. Once if the reviewer asks you that can you give any proof that you did this particular test and you received these results only and there's no manipulation of the data, you can always show them the videos. There are track plots and heat maps also that you can get from this particular software, which you can show to the reviewers or anyone that asks for the validation of it, of the behavioral data that you get. So this is again a video that I mentioned earlier. This is the OFT open field test video of the control animal. The first one is the actual recorded video of the animal during the test, and this is the track plot. You can see the behavior of control rat, that it is exploring all the corners of the arena. It will explore properly. This is the rearing pattern, that when it stands on its two paws. Okay, so the next one is the same experiment for a PD animal model. So you can see the difference between the exploratory behavior of control and animal model. So you can see there's slowness of movement, bradykinesia, anxiety like behavior. The control rat kept on exploring for the entire experimentation duration, but you can see that this rat from the origin point it just went to one of the corners and it will keep sitting there for the entire experiment duration. So okay, so this one is heat map and track plot representation of control and rotanon. The first, the same one for the from the video I just showed you. The other one are the different treatments that I gave in my own study and checked the effect of those treatments in improving the animal behavior. So okay, so these two are the different doses of different tests, different treatments I gave, and these are the standard drug. Okay, so this one is NOR video for control animal. The round object here is the novel object, and the rectangular bottle is the similar object. Okay, so this one is for the PD animal model. You saw in the control rat that it explored both the objects as well as the whole arena, but spent more time with the novel object. And this is the diseased animal. So this animal went to the similar or the known object earlier, and now it will start displaying anxiety like behavior by staying in one corner of the arena, and it will not explore the novel object. So that's all about the behavioral test and anime software. If you have any questions, you can ask about the software or different behavior tests.
How frequent do you do this training phase? Like during training, how frequent do you train? Like in a day, like in a day, how many times do you train? If a police already is like procedures.
That depends on your experiment. So in our research question, we trained them, like we did habituation first. We habituated them to the arena. Then after 1 hour, we did the test. Right. So it is important to habituate the animals to the arena when they will be doing the experiments, but the frequency, that how long we want to keep them training them for that.
Each day.
Sorry, yes, we have to train each and every day separately.
And for Rodeo,
Same for the experimental setup and procedure that remains same for every kind of treatment. It may be control or rotenone or maybe a different treatment drugs that you are testing.
These latest parameters should be all be same for every treatment.
We try to do the test for all the animals in the same particular way, because once you go to another day, your apparatus, because you have to set the anime software each time, you have to give all the parameters or the specifications to the anime software. You have to draw the entire apparatus in the software to basically define the area in which you will be doing the experiment. So that changes a little bit every time. So if you want to compare the results of control to your treatment group or to the animal model group, you have to perform the test on the same day.
Is there any difference in the different tests used to study the same aspect? Like there is the L10 plus case test, there is the ITAR box essay. They are all coinciding. Like why
We
do we
basically validate the result of one test. Because in behavior test there will be various variations that you can see, because the animal won't perform in a similar way in each test. So in one test you might see a particular behavior of the rat that you might not see in another test. That depends on the various different factors that get worked out. If you are performing the test, you perform one test early morning when they are active, or during the night, because they are nocturnal. They will be active during night. If you do during the afternoon or during any time of the day, you will see that they are not performing that well. So you will get different results at different time of day, and various other parameters and factors they also play a role in the behavior of the rats. So basically different kind of tests are available to validate the result of one test to the other, and the ultimate validation is done on the basis of all the other results that you perform after the dissection of the rats.
if you're surgically inducing some cognitive impairment, for example, so there are two groups, there is control and there is sham. So what should be the baseline? Which one should be considered
That
the
depends
baseline?
on your, again, on your model, experimental model and null model. If you in your model, for example, this one, the work that I just told you about, this PD model, I induced the PD with rotenone, right? I did not do any kind of surgery to induce my model. Only if I did any kind of surgery, I surgically injected the rotenone in particular brain region or any other thing to induce Parkinson disease. In that case, I had to use sham. Why? Because in sham I'll be surgical method, but I'll only be giving any kind of buffer or something.
And there will be a control group too, no? So which one should be considered the baseline characteristics, the control one or
I
the
think
sham?
for that you should compare the results with sham, because that will give your the good results in terms that if your surgery is responsible for any kind of behavioral change, if surgery is doing any behavioral change, that will be visible in both sham as well as your other surgical groups as well. So in that case, I think sham should be the baseline.
And while doing behavior experiment, have you encountered any challenges like variations in the result seen?
Yes, in behavior tests there are a lot of variations. I myself performed these tests, I think, n number of times. At first I was not getting any results because as I already told you that different factors play a role. I used to do my test at any time of day. I started doing that in the middle of the day, during the afternoon, the animals.
But again, they are nocturnal animals. If you do the animals during the night, you are doing it for all the group. So even if the performance is reduced, the error, they will be uniform in everyone. Like 4 groups if you are taking, you are doing it at night. For all the 4 groups you are doing it at night. So now ultimately the result should be comparative. We draw the significance on the basis of comparison between the groups. So do you think that is really a factor, the timing of the treatment?
Okay. Timing is one factor, but I think to answer your questions better, the number of animals in particular group, that also matters, because they are after all rodents. They will behave differently every time when you place them in a particular arena. But if you have a larger animal, the number of animals, you are using 8 to 10 animals in a particular group. So there's a probability that more than half of the rats will give similar results, but 2 to 3 rats will always give variable results. So that is why we use higher number of rats and we apply stats for that purpose to minimize the error in the data that we are getting.
If you are doing 10 animals, how many outliers do you usually expect or accept?
Outliers, there may be 3 to 4 animals in a particular test that may perform differently than all the other ones. So for that purpose, you might have to repeat the whole entire group another time. Again, if you are not getting any animals, you may have to change the set of animals. You have to get fresh new animals and you have to perform the test again.
If you are reperforming the test and with the same answers, then what is the validity of it? Because it is based on remembrance. So they have already explored the whole apparatus. So next time they will have some memory of the apparatus and the results will be very different.
Yes, and one more adding to the question. When we have all the established models working on MPTP, then these are all day based. For example, if we take 1 day for, we will take 1 day for to carry out the whole Y-based spontaneous alteration. Then novel arm, if the group is more, it might go till 2 days. Morris water will take 5 days and the probe try will take 6 days. But if we are supposed to repeat whole Morris water again, then we are increasing the number of days of the experiment. So is it good, ma'am? Like now we need the result, we might have to publish. If we extend it, sacrifice day will also get extended, biochemicals and all.
That you have to plan your experiment, I think, because animal tests, they take time. You have to give time to behavior test, and it is not significant to ethically significant to do more than 3 to 4 behavior tests on one particular set of animals, because they will not perform. If you do keep on doing 10 tests on one particular set of animals, they will not perform. They will only give validated good results, the results that you want, in 2 to 3 tests. So choose the best tests that are really important for your study and perform those tests. If you want to do another set of tests, you may have to do the entire animal experimental protocol again and test the other parameters that you want to test, but again the control animals will be different and the model animals will be different, and you will compare the model animals to that set of control animals to the new test that you have done.
Ma'am, in your this period while you were doing your project period, did it ever happen that even after performing twice the behavior, you like had failure and you had to replace the entire set animal?
Right,
It
yes,
happened, right?
many number of times, and I performed behavior animal test. Maybe 2 to 3 years of my study was based on animal test, and I performed again and again the tests because I was not getting. There were different changes that I did to my study. I tested, like I was not getting the results, so I had to change different parameters, test different parameters, that what is the problem that was there in my test. I had to identify the problem, and I had to re-perform the tests to get the ideal results that I should get in comparison to the literature that I saw.
Ma'am, and you do it alone, ma'am?
Most of the tests alone, but in the initial phase I had to get some help
Because ma'am,
because
the in my case, the problem which I've encountered is that while clicking and all, you are setting the animal, then you some time goes while you go and just click in the software, you lose some time in that period. So
yes,
what
you
you have,
have to be fast enough,
yeah.
and you have to you know uh, learn to do the test efficiently, because at the end of the day PhD is your own independent work, and you have to learn to do your test independently. In the initial days even I took some help, but no one helps for more than one particular period, you know that.
Thank you, ma'am.
One more question. After taking all the measurements in a control group, then if in case if we have to do stereotaxical measurements, then after inducing a drug directly to the brain, how much, what are the indications that by then we have to measure all the parameters?
You are saying that after how much time of the surgical
Procedure.
procedure
Yeah.
you have to do the behavior tests.
Yeah.
That is based on the literature. I think you can get better results that in how many days the disease is being induced after the surgical procedure from literature, and you will take that as a baseline. You will perform the behavioral experiments on the last day, that is the period of the what do you say study timeline, right?
Yeah.
So that for that you have to do the literature survey and check that what is the minimum number of days in which the disease will be induced, and you can get the symptoms in
Do
behavior
we have
tests.
to check these parameters every day,
No,
or
no, that every day you can check every day as well, but that also depends on your study, because in my study was for a duration of 35 days, and I checked thrice during that duration. Before the start of the experiments, to check that all the animals are performing like control, right? In throughout through the midway, and then at the end. I did it thrice, and that also depends upon your study design. So you will go through literature review and see how how people are working in that particular area and how you want to move forward with your study. That all depends upon the research questions, your disease model, and the study design.
Did you use one pair as well, and did you have the updated
No, I did
software?
not use Onfray, but one of my fellow lab mate is using Onfray, and she is using in UIPS, Punjab University, pharmacy department.
Right. So
Yes.
that's a like a sophisticated situation where we use the
Right,
prestige
right. It's not
system.
available everywhere, so that's why she's also working in UIPS for behavioral analysis. Any other questions? Thank you.